Good morning.
Good morning, Charlie, and good morning, everybody else.
Thank you for tuning in. Very excited for today's session. I've got loads of questions to ask you, so I'll kick straight off with the first one. Can you tell us a little bit about your career journey and what it is you do now?
Yeah, career journey. It's so I grew up in Cornwall. I was born in Yorkshire, but I grew up in Cornwall. Um, and I think from actually quite an early age, even primary school, I knew I was more scientific than I was artsy. Really enjoyed music, and I still like music, but with all the arts faculty subjects, like English and history, I love English. I like reading books. I enjoy history, but I always got a little bit discouraged when I'd write an essay and the teacher would say, "That's interesting." I go, "Is that right or wrong?" Whereas I really liked understanding the natural world. I liked that there was actually a real answer that you could get to rather than opinion. And I guess, yeah, so secondary school very much oriented towards the sciences, and then did A-level maths. This is going back. Maths for biology, chemistry. Then wasn't sure. I still wasn't sure. Age 17. It's one of the funny things as within the British system that we're asking 17-year-olds to decide their future. I thought I might want to be a medic at one point, maybe a psychologist earlier. When I was 13, I was determined to be a marine biologist. Get horribly seasick, so that was a that was a non-starter. Um, and yeah, then I thought I gravitated towards biology. I guess probably 14 onwards, I could see biology was what I really liked. But even then, I didn't know if it was the sort of more molecular end or the whole organismic end, etc., etc. If you asked me age 17, was I going to be an evolutionary biologist? I've gone, no, not interested in evolution. Largely because I think evolution is really poorly taught these days. Almost became a biochemist, almost became a medic. But in the end, I was fortunate. I went to a university that allowed you to do quite a lot of different subjects in the first year. Going on only really specializing in the last year. And it was through that I discovered, yeah, I'm an evolutionary biologist. And that's what I really love. And then got a very nice fellowship to go to Harvard for a bit. That was nice. That was fun. Then did a PhD back in the UK. And sort of discovered that I don't like complicated problems. I like relatively simple problems. Where I gravitated to do evolution of genes because I thought, organisms are quite complicated things, but genes we might stand a little bit of a hope of understanding. I'm still wrong in that. I don't think I do, but at least I can try my best, as it were. Yeah, and then since then it's been following my interests, following the data, because my field is very privileged at the moment that we are in a second industrial revolution. Gene sequences are being pushed out at an unbelievable rate at the moment. So we can answer questions that were unimaginable before, that we could imagine the question, we couldn't imagine getting an answer, but now we can. So it is a very exciting area to be in. What do I do now? Well, I crunch large amounts of sequence data to ask fundamental questions. The ones I'm particularly interested in at the moment, I guess, you could broadly summarize by looking at something like the human genome and asking, why is it that weird? So if you've done any gene expression within your A-level or whatever, DNA makes RNA, makes protein, and that's sort of the canonical model of what DNA does. But that's 1.2% of our DNA that does the DNA, makes RNA, makes protein. And that's not very much. There is more, about 8% of our genome, of all that DNA, is old retroviruses, things like HIV that are just inserted and died on our genomes. The great majority of it looks as it's dead transposable elements. So why is our genome like that? Where in the genome is the stuff that is important? Why are other genomes not like that? So if you go and look at an E. coli's genome, our laboratory bacteria, oh, it's beautiful. It is so lovely. Every gene is just one nice, lovely gene, and they all have a name, and they all seem to be in the right place and do the right thing at the right time. And 89, 90% of the DNA is protein coding, the bits that aren't the switches for the protein coding. Beautiful, live, lovely genome, and ours just looks like so much rubbish. So So why? But that then turns into another series of questions related to can we understand what bits in our DNA are functional? That's got clear relevance to the problem of diagnosing diseases, genetic diseases in humans. So you will all have been born with about 50 new mutations, somewhere between 20 and 100 usually, depends on the age of your dad. Older dads, more mutations in your DNA. These are new mutations in you. You'll have others from that they inherited that they will pass on to you as well, and so on and so forth. And if you've got a genetic disease, one of those mutations is what's causing it. But many of those we think are going to be just uninteresting, unimportant, but trying to work out which ones are important. And we think about 10% of all the DNA is important, i.e. mutated, it could cause a disease, but only 1.2 is giving you protein. So what's the other stuff? What's it doing? Can we identify it? And evolution is a really good clue to that, because sites that are very slow evolving typically are sites that are important, and important means if you mutate it, you get a disease. The other thing that I'm very involved in at the moment is also gene therapy. So here we can ask, how do our genes actually evolve? And knowing that, can we make a better version of them, a really good version of them to use, give back to people who've got a genetic disease and make sure all the gene works fine and it's safe and so on and so forth. Turns out it's a real problem just be taking one of our genes, going, we'll make a more compact version of it, just put it in a DNA and get it to work. They typically don't. It's one of those other mysteries. Why don't they work? They work perfectly well normally. How come they're not working well now? So yes, it's sort of at the interface of evolution and medicine. That's a lot of what I do. But I'm also very interested in, we do some work in schools and also doing large survey work to understand how best to teach evolution. And the answer is very easy. You teach genetics. Rather, you teach genetics before you teach evolution, and that works. And also, I'm really interested in the problem of why some people don't accept consensus science. I think this is one of the great issues facing the world at the moment, which is misinformation. So why does some people suck up misinformation, other people know to be more critical of it, and so on and so forth? And we discovered a few regularities there. So that's quite fun. Going back to my 12-year-old self, which thought I might want to be a psychologist, so I'm rediscovering that. That's good fun. I like it.
Yeah, thank you for answering that one. And you also answered another question within that, which was why did you choose that? So you covered two within one. So I like
Excellent.
it. So our next question is, how do you stay up to date on all your latest advancements in the field, and how important is it as a scientist to kind of keep learning and keep up to date?
It is one of the great challenges, it has to be said, keeping up to date with the literature. I mean, probably as we're sitting here now, there are thousands of scientific papers being published. And yeah, there are ways and means. I'm quite disciplined. Every Friday, I go onto a number of online websites. I've got a couple of key papers in my field and go, who's been citing these? And that's a really good guide as to the stuff I ought to be reading, and so on and so forth. Keeping up to date is actually, I think, is rather one of the fun bits of what I do. So a lot of what I do, I actually teach myself, but it keeps you really refreshed. I was thinking about this just this morning, that when I started out, I was taught one programming language, which is utterly useless for bioinformatics. Well, that's sort of what I do: biology, informatics, handling large amounts of data, sequence data mostly. And over time, I've learned, I don't know, I've taught myself four different programming languages to be able to handle the data, and they're getting better by the day, and the standards keep shifting. So these days, it's mostly, for those of you who are interested, Python and R are the two big ones in my field. But yeah, so I have to teach myself. I used to, when I was much more disciplined, I used to have a rule that I would learn or do something new every year, just to keep me fresh. 1 year, I have to say, it was learn the trumpet, which did not go down very well in the family. But no, it's learning new skills, etc., etc., is great fun.
I like that idea. Learn one new thing a year, even if it's not to do with your wanted career or and it's something different, like learning a musical instrument. It sounds like a good sort of ideology and mindset to follow. Our next question then is can you share an exciting discovery or breakthrough that your research group has made and why is it exciting?
Oh, now here's a great question. There's been a couple along the way. Oh, God, what an awful question. What can I say? I think, let me go for, so there used to be, let me backtrack this one a little bit. So if you look at the genetic code, we know that there are 64 codons, yeah, that specify 20 amino acids. So lots of our amino acids have more than one codon. I hope the biologists out there understand what I'm talking about. So, for example, if you want to specify glycine, you can do it with GGA, GGC, GGT, or GG. The other one I missed, G. And so we used to think that those third sites were irrelevant. This is actually what I work on a lot at the moment. Those are known as synonymous sites, or they used to be called silent sites, because we assumed that they were silent, that it didn't matter which one you had. And the conventional wisdom in the textbooks was that, certainly in mammals, that it's irrelevant. It doesn't matter which one you have. Now, obviously, for making a transgene, maybe not. And a couple of years ago, we were doing some investigation. We noticed that there was a bunch of proteins that sit actually on the RNA itself. And we wondered, could that actually be imposing selection on the synonymous sites in the RNA and hence in the DNA to say, come stick to me, nice, lovely protein. And we discovered it was the case, and we discovered that actually there really is selection operating on synonymous sites, not by means that people had imagined, but that it really is there. And I was particularly happy with this because we submitted it off to a journal, as you always have to do. And the referees couldn't find a problem with it. But the editor-in-chief said, I'm going to reject it. And I'm going to reject it because it's well known that there is no selection in synonymous sites in mammals. Okay, no, we just showed that there is. And now it's the conventional wisdom that there is. And most of it is to do with these proteins binding onto the RNAs. And it's to make sure that a bit of the RNA is sticky or elsewhere is not sticky for these things and so on and so forth. So yeah, that changed the way people looked at the world, and things that we thought were not important become suddenly important for a completely other reason. And basically, I've sort of followed that one on for the last, I don't know, 20 years or so. And now we're using this very similar insights to go, okay, how would we make the best gene by changing just these silent sites? And it turns out just by changing the silent sites in an artificial gene, so we can do it with something like GFP, which is green fluorescence protein, so it shows you how much protein you're making because it glows. But you can change your orders of magnitude just by the amount of protein, just by changing these synonymous sites, which I think is absolutely breathtaking for things that are supposed to be silent. They're not silent, they actually are extremely important. And that's why we can say, you're going to have a genetic disease because you've got a mutation in one of these things, or we can design transgenes to work better because we know how they're working and what they're doing.
Really crazy that you just discover something new and it changes the way that everybody perceives something. And I guess that's the joy of science, isn't it? You hope that you will find something that will change people's perspective on things.
Yeah, and it really was just sort of following my nose. We'd had before that a couple of funny little signals, very generic signals that all was not well in the world of synonymous mutations, these silent mutations. They did look as though it was selection, but unless you have a mechanism, no one's going to believe you, certainly not in mammals. And so we thought we had the mechanism, and we published a paper after that, which is one of the rare ones I do, where you do every test and you think, if that's what's going on, this is what we're going to find in the test. We do every one, and everyone comes out absolutely right. And that's the sort of thing that goes, ooh, I'm right. I don't expect to be right. I mean, I expect to be sort of following my nose, having hypotheses, but it's the nature of hypotheses that quite often you get it wrong. And
Mm.
that's still interesting. That's really important, because if you think, okay, if my model says this and I see that, then that means there's something wrong with my model. So what
Mm.
is it? So it's an eternal adventure. I like to think about science actually in this whole process a bit like a slime mold. Slime molds, they sort of grow with tendrils all over the place, and all of these tendrils grow out independently. But if they meet, so here's a tendril, and if it meets an obstruction, yeah, so the data says no, it simply retracts itself and you go out this way instead. So that's sort of science. You go, oh, we've got an idea, we head off this way. The data says, oh no, we'll come back here and we'll go that way instead. And so, but it's happening all over the world with scientists all over. And so we have lots of tendrils, it's been drawn back, it's been extended. And we're sort of trying to work our way through intellectual space, trying to work out what is true and what is not true.
Love the slime mold analogy. um,
Science is a multidimensional slime mold discipline.
our final question for kind of this part of the Q&A before we move on to all our student questions then is going to be about managing time and priorities. Science is quite a big topic. You know, there's lots of different niches and lots of different things. And when you've picked one and you're studying, you've got loads of projects going on, different sort of things you're thinking about. How do you manage time and work out priorities that you need to kind of do? What do you do? What sort of techniques do you put in place for managing your workload?
Yeah, I sort of had to learn this actually, and I don't think there's one answer for every person. I see other groups working differently. So why do I do science? Because I enjoy the process of understanding. I enjoy the process of really thinking through a problem. And fairly early on, I got a couple of big grants and some fellows in and so on and so forth. And I had a group that must have been about 8, 10 people. And I started to get worried because at this point I could see I was losing control of all of the projects. Not losing control in the sense of it wasn't good science being done, but I didn't have the headspace to be intellectually engaged with all of them and with the detail to the level that I wanted to. So I never have a group bigger than about 5 or so now, and even that I find quite a challenge because I like to be able to discuss in detail the nitty-gritty of each and every project and really get inside it, because that's why I do science, is to really understand what's going on. So there's that, but then of course there's lots of teaching. I love teaching. I like interacting with my tutees. Often we sort of have quarter an hour one-to-one meetings which turn out to be 2.5 hours long. Can't stop chatting to them, it's great fun. And then there's lots of dull stuff like admin and so on and so forth, but you have to be quite disciplined, I think. And you also have to be quite disciplined to sort of try and stop it spilling over, if you know what I mean. I think when I was younger, I think I made a little bit of a mistake of allowing it to sort of flood into evening time and flooding into weekend time, and that I don't do anymore. I try and contain things so there is some family time and private time and so on and so forth. But yeah, I would not for a minute say it isn't a challenge, because it is a challenge. But at the same time, I find ways of entertaining myself, because that's why I do the job, no matter what I do. I love writing, I love reading. My favourite days are if I can shut the door and do a bit of computer programming and find a really elegant computational solution to a rather nasty problem. That always feels very good.
Great, I like that, and I like that you can identify your limits and go, I don't work well managing a big team, so I only like to manage small teams. And
Yeah,
how
and I see there are other groups that they have the lab head has 40 postdocs and things like that. That's not me. I can't do that.
No, definitely. Okay, so we're going to ask a couple of student questions now that have come in from schools and students, and we have quite a few of them. But
Excellent.
please do keep coming them in because we'd love to hear from you. Um, and we'll go with, oh, let's start with this one, which is, does evolution help with conservation efforts?
Oh, it certainly can. Yes, I've got several colleagues who work on exactly that sort of issue. So at a relatively micro level, so within a population, for example, we can use the tricks we've got for sequence analysis to look at different populations of the same species and go, oh, that little population up there, they're really interesting, they're really different, and they're genetically pretty dissimilar to everybody else. So lose that bunch and you're losing quite a lot of interesting genetic information. So they might say, actually, the bigger bunch seem to all be intermating. They're no threat at all. That bunch over there, however, stuck out on that island, they're actually genetically interesting. So maybe focus our efforts there. Another way that evolution and evolutionary genetics can help is to try and identify on a much broader scale, sort of same sort of principle again, but imagine you've got 10,000 beetles in an area, and you do the phylogeny of them, so you work out the evolutionary history of them, and you go, actually, that lot over there are genetically really different. They're isolated, they're genetically, they look different, they have a different sort of diversity profile. All these others are, 9,990 beetles, none of which can you really tell apart from each other unless you're a real beetle expert. So there, you can use it to identify again species and species groups that look to be that little bit more different, as it were. And then we can also use it in turn to identify populations that we think are more at risk and species that we think are more at risk. So very recently, these classifications of how much a species is at risk. Recently added into that was actually some evolutionary information. So we have a concept which is called the effective population size, which is not the same as the population size, rather strangely. We've got the word effective in front of it. It's quite hard to measure actually what it is, but it turns out in evolutionary terms, it's a really important number. Because if a species' effective population size is rather low, it means they're much more likely to be under quite imminent threat of extinction. And that can happen because, for example, if a species is highly inbred, you can get a low effective population size. It could be that not many of the males, there may be a sort of 50-50 sex rate, but not many of the males are actually reproductive males. And that will give you a low effective size. Most of those males are sort of redundant, as it were. And so that is now actually that number, and calculating that number is now in one of the checklists for identifying species at threat.
Really interesting to think about the wideness and the variety that it can be applied to, and how important genomes are, and kind of genetics and things, and helping us identify all sorts of different stuff. It's really interesting. So thank you to whoever asked that question. I will say the question came from the heart of Worcester College, so thank you for sending that one in. Our next question then, and we've had a couple of questions that have come in from Uxbridge College, really interesting, and they've asked, how are traits inherited? And is that connected to genes?
Oh, absolutely. So there's a whole large area of genetics, often closely coupled to evolutionary genetics, which is called quantitative genetics. And quantitative genetics is concerned with exactly this problem. How is it that phenotypes, traits are themselves inherited, particularly in the context where, if you take something like height, for example, there is no, Mr. Mendel's got very little to say on this issue, because Mendel was working one gene, two different versions, and you get yellow or greeny seeds, and you get wrinkled and unwrinkled, and that's one gene, one trait. Those we've known about for quite a while, but something like height, hundreds, if not thousands, of genes and the variants within them will be contributing to the variance that we see between individuals in height. But of course, it's not just the genes, it's also the environment. So if you go to the Netherlands, for example, you'll notice that on the average, people are a bit taller, and that is probably not much to do with the genetics and everything to do with a very dairy-rich diet, particularly in the early years. So we have these environmental components and we have genetic components, and the field of quantitative genetics is saying, can we work out which genes, what the contribution of the genes is, what's the contribution of the environment, and so on and so forth. And that gives you concepts like heritability. And heritability is a number between 0 and 1 to say all of the variation that you see in a population is owing to the genetics. So if somebody is tall versus somebody is short, all of that variation could be, that's heritability 1. is owing to genetics. Naught means it's nothing to do with genetics. All of the variation is environmental, and so you get a number for any given traits between the two. And some traits, it turns out, are highly heritable, and some traits are not particularly heritable at all. There's a number which look as though they're probably not heritable at all. So in fruit flies, for example, the well-known fruit fly, you'll find individuals who put their right wing when they're resting, they have a nice rest, they can put their right wing over their left wing or their left wing over their right wing. And it turns out, as far as we can see, this is not heritable. So you mate a lefty-lefty with a, sorry, left over left, sorry, left over right, I'm getting myself confused here, left over right with a left over right, or a right over left and a right over left, and then they produce the same ratios of lefts over rights and rights over lefts. So as far as we can see, that's one of the rare things that appears not to be at all heritable. But height is pretty heritable, we think. We think the heritability is about 0.8 for that. So 80% of the variation between individuals within a population is genetics. 20% is having lots of cheese when you're a child.
Interesting. Calcium does make your bones go stronger.
Calcium absolutely helps your bones grow.
So yeah, really, really interesting. And I'm sure some people are going to be trying to figure out and Google what traits are more heritable than others now, because that's a really sort of interesting concept. Our next question then comes from Notre Dame Catholic College, and they have asked, how will AI impact your research? Or does it, is a nice other way. So how does it, or does it affect your research?
It does. We're actually doing some AI type stuff at the moment. Basically, it's just blind statistics. So the conventional mode in which you test a hypothesis is you'd start with, oh, I wonder if it could be this, and then you'd modify it and you see if it correlates and so on and so forth. And that's sort of doing one thing at a time. But particularly within genetics, so many things are so complex with such complex interactions that actually there may well be limits to that let's do it one at a time approach. And what AI allows you to do is a sort of blind way of going. And so we're actually using AI to try and work out which synonymous mutations may or may not cause disease. And we can do that by going, here's a bunch that we know do, here's a bunch that we know don't. Try and get AI, and then you feed it. You can feed it 1,500 different numbers about the ones that do and the ones that don't. And then basically what AI is doing is a sort of dumb statistics. Can we work out patterns within this as to which ones do and which ones don't? And so you can have a training set and you go, okay, well, let's apply it to a bunch of others where we know, but let's see if it's worked them out. So we had the first go at that, and it turns out that you don't need 1,500 numbers, you only actually needed 3. And so with just 3, we can have really quite striking accuracy. So actually the old approach, just take these 3 parameters and go, okay, we can now predict which ones do and which ones don't. And adding in any more just doesn't really add very much. But that might be because the other ones that we added in weren't the right ones to add in. Which is then always a problem, because you do sort of need to think, well, what could be important to add those into your models. I'm personally rather reluctant to go down that line, not least because it is a sort of dumb science, if you know what I mean. It's quite a hard program, but what's coming out is you're letting the machine do the thinking for you.
Yeah,
Well, I'd like to do thinking.
That's sort of that
Taking my
worry,
job
isn't
away
it?
from me, you might say.
Yeah, that's the worry that some people have, that it's, you say it's freeing you up to do other stuff, but you might enjoy doing that little bit as
I
well.
enjoy working it out, and then whatever comes out of it, you go, why on earth has it found that parameter? That's weird.
Mm. Our next question then comes in from Barking and Dagenham College, and they've asked, what jobs can I work with a science degree?
Oh, world is absolutely your oyster with a science degree. So I mean, let's talk about my tutees here at Bath, and they go on to just about anything. Some of them discover that there's a certain sub areas of science they particularly like, and so they'll do a master's afterwards to retrain, then academia, industry. So a good number of my graduate students have gone to Genomics England, for example. They're programmers for Genomics England. A good number discovered that they really like communicating science rather than doing science. So two of my ex-lab members, one became science correspondent for The Guardian, another for BBC Focus magazine, for example. And I'm perfectly, I think science communication is really important and should be done by good people, as it were, who really understand the science and can write and can communicate. And so happy to give them training in those writing skills as well. They write much better than I do now, it has to be said. But some go, no, this isn't for me. I've got some who, gone into marketing, particularly marketing for science-related industries. But I had a colleague who went into law, rather interestingly, not so long ago. And that was to become a patent attorney because they really need the scientific knowledge. And then, of course, some go, I just enjoyed science at university, I'm going to do something completely other. So quite a few go into the civil service, etc., etc. The world is absolutely your oyster. I think a good science degree should give you some good technical skills, as well as lots of good soft skills like presentational skills, writing skills, and so on and so forth. But yes, I don't think a degree in science would stop you going anywhere. You may not. It may be quite an achievement to become an academic in medieval history, having done an undergraduate science degree. Not sure anybody's ever done that, but you should. Other than that, no, that it stops you doing nothing and enables you to do loads.
Great. Our next question then is a science-focused question. Really interesting. It's come from Dooley Sixth Form, and it's: do you believe that it's possible that genetic engineering could be used in the future?
It's being used now, or do you mean on humans?
I think on humans.
Oh, on humans, right? We'll stick to on humans now. Okay, so we need to distinguish a couple of things, because at the moment there's a sort of genetic engineering which is being used. So gene therapy is in effect a form of genetic engineering, and I don't know if you've heard about CRISPR and gene editing, but that's now being used. For example, as of a few months ago, both the US and the UK approved a CRISPR gene editing approach to cure sickle cell disease, a very common disease both in Africa and America. And gene therapy is, in a sense, a form of genetic engineering. So, for example, there's, again, within the last year, there's been a licensed version of a gene therapy to cure haemophilia. And notice these are cures. These are not therapies in the sense that you take a drug and you have to keep on taking it. So something like haemophilia, for example, the problem is we could keep it at bay. So haemophilia is one of these bleeding disorders. And in some cases, you need a transfusion every couple of days just to be able, if you can't make the protein, factor 9 and factor 8 are the two typical ones. If you couldn't make it, you need a blood transfusion, or there are artificial versions of the two factors that you can be given, but you have to keep on being given them, and that's every few days, and so on and so forth. With gene therapy, we give you the gene, and the gene makes the protein for you. So in that sense, it's a real cure for the condition. And in this sense, that is genetic engineering. What we're doing is we're engineering a right version of the gene, and we got very clever technology now to get that right version of the gene into your cells and get it to work. So you were born with a non-functioning version of the gene. We're giving you the well-functioning version of the gene, and they seem to be working quite well. Now, where that's not classical genetic engineering is that this is not so-called germline therapy. Now, in germline therapy, what you do is you wouldn't just change your body, you'd change your offspring as well. So giving individuals gene therapy for sickle cell anemia, for the haemophilias, and what have you, a couple of eye disorders can now be cured as well. Giving that, it does not stop your kids from getting the same disease. So if it's in your DNA, it doesn't stop your kids from having it. So to stop your kids having it, you'd have to do so-called germline therapy, or you'd work on the very early embryos, and you'd modify the very early embryos, put them back into mum, they would grow up, and then it's sort of hard fixed into the DNA for all generations down the line. So that is very, very controversial. So you may have heard that there was a Chinese doctor, Dr. He, um, who claimed that he'd done this. It looked as though he did do it to modify a gene called CCR5. This is involved in HIV resistance. So his argument was modify the embryos so the embryos themselves would be HIV resistant. And it caused quite an outcry because that was modifying the embryo, so all the offspring down the line would then also have this change. And it caused an outcry because we don't think we're ready to do that. We don't know what the side effects of this would be, particularly with something like CRISPR gene editing. You can have what are called off-target effects. You think you're modifying this thing, but you may be modifying something else. On top of that, we don't really understand what CCR5 is doing in this particular case. It seems to have effects in brains as well. So the fact that it's giving resistance to HIV is very nice, but who knows what you've done to these folks' brains. But then on top of that, it is actually not the case that, and this is where the ethicists were particularly concerned, HIV is not something that is not curable at the moment. It's not a serious, not curable disease. HIV is now effectively a well-handled disease. So they couldn't see a case for germline editing. So at the moment, there is a moratorium on germline technologies applied to humans. Cannot guarantee that there's nobody in the world doing it at the moment just because there's a moratorium. But yeah, that would be very contentious if you ever heard about that. But the using genes to modify your DNA to cure your diseases, the last count I saw at NIH, the big American, and the FDA, sorry, approving these things. They're expecting a couple of thousand applications for gene therapies for this disease or that disease in the next few years.
Very interesting. And you answered a couple of more questions within that they'd asked, which is
Amazing.
really great. Yeah.
I'm psychic and I can understand questions before I'm even given them.
They did pose one other interesting question. I'm going to ask it. It's kind of a hypothetical question. They asked, if we know which genes do what, why can't we build a species?
Oh, to a first approximation, we can, and we have. By which I mean the gene sequencing, no, gene synthesis technology. So sequencing, just you take DNA and you can work out what the sequence actually is. Gene synthesis technology and genome synthesis technology, we can take the raw materials of DNA, glue them all together, and you can make a designer genome. I'm sure it's what all the fashion conscious people will be wearing very shortly. But it's been done. So you can do this with small bacterial genomes, for example. You go, this is what this genome should look like. I'm going to make a synthetic version of it, put that back into a bacterial cell, and yeah, you can make a brand new species like that. It's working up at the moment. There are getting to the scale of the human genome is a challenge, but we can do it on simple eukaryotes at the moment. Not something I do. When I say we, I mean everybody else, not me. But it is something that can be done. People are thinking about doing at a sort of at a grosser level. It has potential applications in quite a number of areas. It should tell us an awful lot about how things function as well. So we could make a DNA and go, well, we're going to leave out all the bits that we think are not interesting. Do you still get a well-functioning organism? What an interesting question that is. Now, I'm not suggesting we do that with humans. Let's not go there. But certainly with something like yeast has got 6,000 genes, etc. It looks to have a few sort of wibbly wobbly bits towards the end of the chromosomes we don't think do very much. And in principle, we could just go, okay, here's a really nice simple yeast genome. It's got its 6,000 genes, all the bits that we think are important. Does that actually make a functioning yeast?
Very interesting question. Thank you who proposed that. We're heading towards the end of our time, so I'm going to ask our last question, which has come in from the Greycoat Hospital School, and it's some say that a young person is really interested in pursuing a career in science. do they need to get any work experience? And if so, what type of experience work experience would you recommend that students who want to go into the sort of field that you're in try and get?
I think there are two different questions there. I think within the British system, there's something odd about the British system, which is that we ask 17-year-olds to make really big decisions. And so I would say one reason to have work experience is not so you have a line on the CV. It is actually for you to discover, is this actually something I think I could enjoy doing? Is this something I'd want to get out of bed in the morning for? Rather than the line of the CV. So the second reason is the question being asked, is somebody going to look at me better if I can say I've got some work experience? Probably. I don't think it makes a particularly big difference. But in terms of a difference for your thinking, I would always say, try and work out what you think you might be interested in, and then go and find somewhere relevant and see, am I actually interested in this? Because I think there's a big dislocation between what people think science is like and what science is actually like. So it's not uncommon, for example, I have plenty of tutees who come in their first year saying they want to be research scientists and they're going to be lab scientists and they're going to, great, I'm not going to discourage them on that. And they, about 78% of our students do a year-long placement here at Bath. And many of those are lab placements, and it's not uncommon that they come back and they go, I like science, but I can't do science. Because I think the brain for science and the temperament for science are two different things. So you can be as smart as a cookie in being able to do science and think about science, but the physical process of doing science is breathtakingly slow. Things frequently do not go right. And the rate of progress is, horrible. So why I'm actually a bioinformatician. In principle, I can actually get the answer I want within no time at all. But I usually have a rule that if you think how long an experiment should take in molecular biology, approximately multiply that by 5 and add a bit. And that's more realistic. So if everything went absolutely perfectly, your experiment should take a month. Realistically, it's going to take 6. If it should take no more than 2 months, realistically, it's going to take you a year. With my graduate students, I start getting worried as soon as we go over the ×6 mark. I think, oh, this is probably a little bit too long now. Maybe time to drop this. It's not working. But yes, having the temperament to be able to work at that sort of pace, and then check and double check, and then you're doing your PCR and somebody's contaminated the water, so you've got to do it all again, and the bands aren't clear and the journal doesn't like it, etc., etc. It's quite a frustrating process. So yeah, go and shadow, go and do some work experience, but find a field that you think you might be interested in. And use it much more for your exploration of the areas that you might be interested in, rather than, oh, here's a line on the CV.
Well, we are at the end of our time. So do you have any final thoughts that you'd like to share with our viewers before we end today's session?
I guess one thing we haven't talked about is how exciting. I don't think people recognise, particularly the biology A level as it is, I don't think they recognise how exciting biology is these days. So here's a couple of funny little facts. So if you take every letter in every book as one piece of information, it turns out that, and on top of that, we have another problem, which is that every base pair in DNA that's ever been sequenced is one bit of information. All good. Then 98% of all the information humans have ever accumulated was accumulated in the last 2 years. Isn't that amazing? It's nearly all sequence data. And in no small part, that's because the timing and the price of sequencing is just astronomically low. So when the first human genome came out, 2003, it cost just under $3 billion, took about 15 years. It's now doable under £100 and is done in real time, i.e. as you wait, the sequence is there. And so there are cases now where people have turned up at hospital, had their DNA sequence, genetic defects identified, cure suggested, cure active, and that's all been done in 2.5 days. It is amazing. We are revolutionizing. The DNA sequence data is revolutionizing every field of biology because it's so cheap, so abundant, and the real skill sets are now much more analyzing it and making sense of it, which is what I do. I happen to find myself in the right place.
Yeah, perfect for curious people who are excited and want to make a difference.
Absolutely, absolutely.
Well, thank you for joining us today, Laurence. It's been wonderful having you on, and yeah, thank you for giving your time to talk to us.
My pleasure.